You may remember, you learned in grade 7, that there are different kinds of unicellular organisms in pond water, and hay-infused water samples, when they are observed through a light microscope. The organisms, which are invisible to the naked eye, and which cannot be seen other than through a light microscope, are known as microorganisms. In this lesson, we are expecting to learn about a few types of microorganisms, which live in our surrounding environment. The invention of simple microscope by Antony van Leeuwenhoek, a Dutch scientist in 1674, can be considered as the beginning of the exploration of microorganisms. The compound light microscope is used to observe very small objects, which are invisible to the naked eye. Although microorganisms cannot be observed with the naked eye, or a hand lens, they can be observed through a compound light microscope. You already know that the compound light microscope has been made using two convex lenses with high magnifying power. Now, we will try to observe a sample of coconut water, which was kept open for 3 days, using such a light microscope. For this, a clean glass slide, and a cover slip, are required. Place a drop of this coconut water carefully, on the glass slide, and cover it with a cover slip, and then observe through a light microscope, under low power. Then, it can be observed that, there are many organisms that cannot be examined with the naked eye. Do you know, that this unicellular species is a type of microorganism, known as yeast? According to this, the single-celled, or multi-celled organisms, which cannot be observed with the naked eye, when taken individually, are known as microorganisms. Microorganisms have adapted to live in all places in the environment. For examples, microorganisms live in the atmosphere, water, soil, on, and in living organisms. Not only that, some microorganisms also live in glaciers, deserts, and deep sea, where ordinary organisms cannot survive. Further, they are also able to live in hot water springs, saline environments, etc. Microorganisms show a tremendous diversity in their morphological and feeding mechanisms. Rod-shaped bacteria, and also spherical bacteria, spiral-shaped bacteria, comma-shaped bacteria, are examples for microorganisms with different morphological features. Also, different shaped fungal species, unicellular amoeba, paramecium, and a type of algae, named Chlamydomonas, fall in the group of microorganisms. Microorganisms possess different feeding mechanisms. For example, a type of algae named Chlamydomonas is an autotrophic microorganism, and it produces food, which they need for itself. But, amoeba makes pseudopods, by stretching out its body, and encircles other microorganisms, that come nearby, and then eat them. The paramecium makes movements in certain pattern with cilia, which are around its body to create some currents in water, and eats those microorganisms, which are trapped in that water flow, and swept towards the paramecium. In addition to this, Plasmodium also belongs to the group of microorganisms, which is a type of protozoan, which lives in some species of mosquitoes. These microorganisms enter human blood, with a bite from such mosquitoes, and cause malaria. Fungi and bacteria species help to decompose plant and animal matter by living on them. This contribution plays an important role for the sustainability of environment. Microorganisms can be classified to 5 main groups. They are algae, fungi, bacteria, protozoans, and viruses. If viruses, which consist of a very tiny structure, which is not only invisible to the naked eye, but also through a light microscope, are discussed under microorganisms, there is no confirmation yet as to whether they are living or non-living. They show both living and non-living characteristics. Viruses can only be observed through an electron microscope, and viruses replicate rapidly within living cells, and produce a large number of viruses. Therefore viruses are considered to be a living species, because of this reproduction characteristic. The eventual result of virus replication in living cells is the death of the living cells. Therefore, viruses cause very harmful effects to living species. The diseases, such as common cold, chickenpox, dengue, and AIDS, are caused by viruses. There are no medicines to treat viral infections, however antibodies, which are created within the body, can cause natural healing for some of those diseases. Some viral infections can be cured by weakening viruses by these antibodies, however AIDS cannot be treated in this way, and it will finally cause death. In this lesson, we learnt about: there are organisms, which are visible to the naked eye, and invisible to the naked eye, and examples for different types of organisms, which are invisible to the naked eye. You may remember what you saw when opening which was left on the school table with some previous day. You may also remember that it smelled bad, and its color and nature had been changed. How did the food that was fresh previous day become unsuitable for the consumption in the following day? What is the factor that caused this? What are the measures that can be taken for controlling food spoilage? We will study these facts in this lesson. You already know that food which is open to the environment becomes unsuitable for consumption with time. Food becomes unsuitable for consumption like this, as a result of the growth of microorganisms on food. We can conduct a simple activity to observe the changes that happen during microbial activity. For this, let's get a few selected food samples, and observe their change of color, texture, odor, and appearance with time. Accordingly, the changes that take place in those food samples, after 24 hours, 48 hours, and 72 hours, from the fresh condition are tabulated below. According to this, you will observe that the slice of bread, which was white in color with a porous texture, a sweet odor, and a pleasant appearance at fresh condition, is turning with time to an unpleasant appearance with black structures, a brittle texture, and a foul-smelling odor. When you study the given table, you will see that the other food samples are also turning gradually unsuitable for consumption, changing their color, texture, odor, and appearance. Those food items change their color, texture, odor, and appearance, as a result of microbial activity on them. As a result of this process, the nutritional value and the taste of food are changed, and turning the food unsuitable for consumption, since properties of food are changed, is known as spoilage of food. Hence, the main factor causing food spoilage is microbial activity on food. Here, it is said that food has acted as a substrate for the growth of microbial activity. Now, we will do an activity to identify the types of microorganisms that grow on food, turning them unsuitable for consumption. For this, take a small slice of bread, some water, a Petri dish, an inoculating loop used in the laboratory, a glass slide, and a cover slip, a spirit lamp, and a light microscope. Now, keep the slice of bread in the Petri dish, and spray some water on it. Keep this exposed for the environment, for 3 days. After about 3 days, some white fibers, and patches with black structures, can be observed on bread. Now take the inoculating loop, and heat it up well using the spirit lamp, and then allow it to cool down. Here, the inoculating loop is heated up, and cooled down using a flame, to destroy microorganisms on it, if there is any. Now take a small part of white fibers, and black structures on the slice of bread, and place on the glass slide slowly, using the inoculating loop, which was heated up and cooled down. Then put a drop of water on it, and cover the slide with a cover slip carefully, without leaving any air bubbles in the middle. Then observe this temporary prepared slide, through the microscope under the low power. You will observe that there is a large number of white fibers, and black structures on bread. This type of microorganisms that bread unsuitable for consumption, is a kind of fungi, which is commonly known as mold on bread. When a microbial activity takes place on food, it produces various byproducts, and as a result, physical properties such as the taste, odor, color, texture, and the appearance, and chemical properties of the food are changed. When a microbial activity takes place on different food items, it produces various byproducts. Let's conduct another activity, to understand this. For this, take some sugar. Some yeast bought from the market. Warm water of about 40 Celsius. A bottle. A suitable container. A balloon. And a spoon. Add 2 teaspoons of sugar, 1 teaspoon of yeast and 200 milliliters of warm water into a container, and mix well. Observe the nature of the mixture, after leaving it for about 20 minutes. Then, it can be observed bubbling, in the solution prepared mixing yeast, and sugar, and the container feels warmer than before, when it is touched, and the solution smells with the odor of alcohol. You may remember, we learned that yeast is a kind of microorganism. According to that, you can understand that the microbial activity of yeast produces a gas, makes the container warm, and makes solution smelling the odor of alcohol. The production of a gas due to this process can be confirmed by allowing this microbial activity to take place inside a glass bottle, and fixing a balloon to the mouth of the bottle. For this, take 2 teaspoons of sugar, 1 teaspoon of yeast, and 200 milliliters of warm water, as used in the previous activity. Add them into a glass bottle, and mix well. Then, fix a balloon to the mouth of the bottle, and leave it for about 20 minutes. Here, you can observe that bubbling produces a gas from the sugar solution, and the balloon fixed to the bottle gets inflated gradually. Hence, you could understand that a gas is produced due to the activity of yeast on sugar solution. As a result of this gas emitting, balloon inflates. The gas produced in this process is carbon dioxide, and the reason for the odor of alcohol is the ethyl alcohol produced in this process. The carbon dioxide gas plays a very important role in the bakery industry. The activity of yeast is very useful in the production of bakery products, or wheat flour-based food items. Here, the dough prepared by mixing wheat flour, yeast, and flavors rises with time, and the reason for this rise is the carbon dioxide gas produced during the activity of yeast. This is known as rising, and yeast is known as a raising agent. The carbon dioxide gas produced here provides a soft and porous nature to bakery products. Ethyl alcohol produced during the process evaporates during baking in ovens. When food is exposed to air, microbes act on them very easily. The reason for this is because the environment provides suitable moisture and temperature for their growth. Therefore, if a moist food item is exposed to an environment with favorable temperature, microbial growth would take place rapidly. Now, you will understand why food gets easily spoiled in normal room temperatures of 25 to 30 Celsius, and food spoilage is minimized at relatively low temperatures in refrigerators. Hence, you will understand that moisture and temperature are the main factors that contribute to the microbial activity. When there are favorable moisture and temperature, the various byproducts produced by microbial activities change the taste, odor, color, texture, and the nutritional value of the food. Microbial processes have been named, depending on the type of food. Microbial activity on food high in sugars is known as fermentation. Microbial activity on food high in protein is known as putrefaction. Microbial activity on food high in fats is known as rancidity. The factors such as a substrate, moisture, suitable temperatures, and in addition to that, pH range of the medium also affect the microbial activity. Microbial activity can be controlled by controlling these factors. In this lesson, we studied about different effects of microorganisms on food, and measures that can be taken to control microbial activity on food. Let's identify uses of microorganisms for different industries. Humans have been using microorganisms for industries since ancient times. Producing compost. Producing biogas. Coir industry. Producing vinegar and alcohol. Producing vaccines. Producing antibiotics. Producing yogurt. Producing cheese. Producing curd. Producing bakery products. Use as a food are some examples for this. Compost can be produced by collecting decomposing plant matter and things that are thrown away from kitchens. By supplying a good ventilation, microbes grow rapidly on them, and decompose the waste, producing organic fertilizer. Organic fertilizers produced like this, are highly used in agricultural lands. Because of this, it has been highly popular as an industry today. Dead animal and plant matter is decomposed by the activity of microbes on it, and collected to the environment with time. This decomposition process by microbes is known as microbial decomposition. If there is no such process, the collection of such matter will affect the natural balance of the environment. This decomposing process adds different nutrients to the environment, and it contributes to the well-being of soil. Microorganisms are used to control various pests in the environment. Pests are defined as species, which cause harmful effects on agriculture, stored food, or different animals including humans. For examples, insects, different kinds of moths, and some worms, that are living in the environment, can be considered as pests. These pests can be destroyed by targeting different stages of their life cycle. Destroying pests by microorganisms without using chemicals is known as biological pest control. The eggs, larvae, and mature stages of the pests can be destroyed by the action of the corresponding microorganism on them. For an example, by this time, Australia is successfully using special bacteria to destroy larvae of dengue mosquito. Under harmful effects of microorganisms, we will study about food spoilage, various infectious diseases, damage to clothes and wooden furniture by growing on them. We studied in the previous lesson about food spoilage caused by microorganisms. You may remember we studied that fruits, vegetables, and also cooked and non-cooked animal and plant food are turned unsuitable for consumption by microbial activities on it. Different infectious diseases caused to humans by microorganisms can be studied under four main categories. They are diseases caused by viruses, diseases caused by bacteria, diseases caused by protozoa, diseases caused by fungi. First, let's study about diseases caused by viruses. There is no medicine for diseases caused by viruses, however, some of these diseases are cured by antibodies formed within the human body. Some of these antibodies stay in the human body, and prevent reoccurrence of those diseases, but the effect of some antibodies may disappear after some time. For an example, the antibodies formed by viral diseases like chickenpox, measles, and mumps, stays within the body, and therefore the probability for reoccurrence of these diseases are eliminated. but the antibodies formed by diseases like cold and dengue stay in the human body only for a short period of time. Therefore, probability for the reoccurrence of these diseases is very high. Vaccines for polio, or often called infantile paralysis, are given when young, and these antibodies stay in the body, and prevent from polio for the rest of the life. Acquired immunodeficiency syndrome, or AIDS, is a disease caused by a virus, called human immunodeficiency virus, or HIV, and there is no treatment to cure this disease, has been discovered yet. Tuberculosis, typhoid fever, and leprosy can be given as examples for diseases caused by bacteria. The BCG vaccine is used against tuberculosis, and it is given to newborn babies within 24 hours from their birth. The bacteria which causes typhoid fever is mainly spread by food and dirty water. Leprosy is a bacterial infectious disease that causes dissolving muscles leading to separation of finger joints in hands and legs. Since there are successful treatment methods, these diseases can be completely cured by early identification of symptoms and getting timely treatments. Protozoa is a kind of unicellular living species, and some of them cause diseases to humans. Malaria, Leishmaniasis, and Amorbiasis are some examples for this. Among them, the protozoa which spread malaria lives in mosquitoes, and it enters human blood with a bite from these mosquitoes, and infects. By now, Sri Lanka has been successfully able to control the organism, which causes malaria living in human blood. Leishmaniasis disease is not found in Sri Lanka, however, it is found in America. This disease is caused by a protozoa, which lives in insects called sandflies, entering the human blood. Skin sores and severe fever are identified as symptoms of this disease. Amorbiasis is caused by a kind of parasitic amoeba, called Entamoeba histolytica, entering into the human alimentary canal. This microbe lives in the human intestine, and absorbs nutrients. And the toxics excreted by them cause diarrhea. This disease can be even fatal, if it is not identified and treated. Fungi also cause different kinds of diseases. Eczema and pityriasis are examples for them. Microbes cause diseases to animals, as well as humans. For an example, hydrophobia, or mad dog disease in dogs, is caused by a virus, and a bite from these infected dogs can spread this disease to a person. Therefore, it is very important to give hydrophobia vaccines to domestic pet dogs and cats at the proper age. Further, it is important to immediately consult medical attention from the nearest hospital, in case of a bite from dogs and cats. Foot and mouth disease in cattle is also caused by a virus. The infected cattle must be immediately separated from the crib. Otherwise, it can spread to other cattle. It is possible to give vaccines to prevent cattle from this disease. Mastitis in cows is caused by bacteria, and it can be cured by giving antibiotics. Leaf curled sickness in chili plants, and mosaic disease in papaya trees, are spread by viruses. If any infected plant is identified, it must be removed from the field, and burnt out. Potato blight is caused by a kind of fungi, and a suitable fungicide can be used to prevent this spreading within the field. Microbes affect non-living things, as well as living species. Black fungus on clothes, green or black stains on walls, can be given as examples for this. In most of the cases, the beginning of this process is caused by the moisture of the surface. The stains that occur in the presence of moisture are mostly caused by fungi. When fungi and the microbe form of algae are present together, they appear in green. The damage of this microbial activity causes adverse effects on the economy. There are various coatings and paints manufactured today, in order to minimize the effect of this microbial activity. The damage can be minimized because of the water-based and fungicide properties. In this lesson, we studied about beneficial effects of microorganisms, and harmful effects of microorganisms on living species, including humans.